Okay, I think we'll begin. Uh, those of you who were here last week, we all heard an amazing two talks, but the second talk by T.K. Jong was really, I thought, spectacular in terms of information and communication. How's that? Okay. So at any rate, our second speaker last week, T.K. Jong, who gave an incredible talk about HIV, unfortunately died the following day. And an enormous loss to everybody. He'd been here for 25 years and was one of the world's leading retrovirologists. So today we're going to have a session on hepatitis B virus infection, linked in the bridge idea to T-cell biology. And originally Ron Germain was going to discuss the latter part. For personal reasons he was unable to come, but we were really most grateful and appreciative that Michael Lenardo has offered to speak and talk about a slightly different topic, but nevertheless very much related to this general theme. So for those few of you, I think, who haven't been here before, the whole point is to get everybody excited about what's going on both sides of the bridge. For those who don't know, this is the Brooklyn Bridge, the probably most famous bridge anybody can find. And the two sides being clinical disease, therapy, diagnosis, and so forth, and the other being fundamental mechanisms which may immediately relate to the disease, but on the other hand may set directions in which bright young people looking for exciting careers may be interested in pursuing. So, okay. So thank you. So when I think about hepatitis B, these are the things that immediately come to my mind. And I think for those who don't know, this is one of the great classic stories of serendipity and of solving a major health problem. Hepatitis virus infection is one of those 5 different viruses, sort of a global set of diseases that were part of the great epidemics in history and had an amazing story. And what little was known back in the 60s and 70s is that there was one infection that seemed to have a short incubation period and another one that had a longer one. That was about it. The serendipity, which I saw is in my grandson's high school biology book. There's a picture of the famous Octoloni plate where serum from an Australian Aborigine results in a precipitin line when run against the serum of a multi-transfused patient who had hemophilia. The original idea was that this was some genetic polymorphism, but through an amazing series of events, it really turned out that this was the first indication of a true hepatitis virus. And the precipitin line was caused by an antibody in the hemophiliac blood and a virus, a DNA virus in the Aborigines' blood. This work was done here at the NIH. And in 1976, Barry Blumberg, who had moved on to Philadelphia, won the Nobel Prize for this. Now, much of the work was done by one of his postdoctoral fellows, Harvey Alter, whom you have met on several occasions. Harvey comes regularly. I think he's in Jerusalem today, but he's going to speak later in this course about another type. But Harvey and his group are largely responsible for cleaning up much, not all, of one of the big risks, which was getting hepatitis after a blood transfusion. I think the figure, well, Jay will mention it, but it's somewhere maybe around 30–40% of people who got more than one blood transfusion developed hepatitis. It was pretty serious stuff. And with the knowledge of the antibody and of a way of detecting the virus, it was now possible ultimately to measure both virus and antibody and answer some of the age-old questions, like why is primary liver cancer common in some parts of the world, the Orient, Sub-Saharan Africa, and it used to be very rare in the United States, largely until the Asian immigration came of people who were carriers of HBV or HCV. So you'll hear more about this. So one of the focuses is, how does the virus infect? And we're going to hear something about that, or at least what the state of the art is. How does it cause hepatocellular damage? Does it do it by itself, or is this basically an immune phenomena? And what is the role of the immune system on one hand in defense against the virus, the antibody against viral components, or causing disease? Maybe it's T-cell activation. So what's the role of the immune system? And then from a more, I guess, sociological medical point of view, there is a very effective vaccine. So why does the disease persist as a major global health problem? Can we prevent it? Can we treat it? So these are the things that flow through my mind, and I only put them up there because I trust that you have your own ideas, and we'll have an opportunity to hear from two experts about this and other things, and also to engage them in questions, and don't hesitate, if there are questions, even during their talk, to raise your hand or say so. I would ask you, if you ask a question, to speak loudly, because there are several hundred people who are online, and they complain to me when they can't hear the question. So our first speaker today is going to be Jay Hoofnagle. Jay is one of the leading hepatologists in the world in studies of hepatitis, particularly hepatitis infection. He's the director of the Liver Disease Research Branch of NIDDK, has been so since 2003. Graduated from Yale Medical School, did a residence, came here as a fellow, and did some clinical training elsewhere in the VA in Washington, and finally came back to NIH in 1978 as an investigator, and has been here ever since. And Jay is truly the dominant person in the NIH's activities regarding global hepatitis, as you will hear. Now, our second speaker is Mike Lenardo, who's chief of the molecular development of the immune systems section in NIAID. So Michael graduated, got a medical degree from Washington University, and then was a fellow with David Baltimore for 4 years at the Whitehead Institute before coming here. And he's been here as an investigator, and then since '95, the senior tenured investigator. I couldn't help but notice, and probably I'm not the first one, but I'm looking at his awards. I noticed this one remarkable one about one conferred on you by the Queen of England. So maybe you'll comment about that en passant. But so both of our speakers have had really distinguished careers. Mike Lenardo was involved almost from day one in the discovery and proof of effect of NF-kappa B and other factors. So, at any rate, enough of me. Jerry, thank you.
Thank you, Wynn. I get to talk about hepatitis B, which is a disease on its way out. I'm going backwards, too. There we go. Okay. A challenging and an ancient disease that's been found in mummies. It's due to a small DNA virus. It's perhaps the smallest virus that infects humans. It can cause acute hepatitis, acute liver disease. It can cause chronic liver disease. It's the single major cause of cirrhosis in the world, the single major cause of liver cancer. In America, not, but elsewhere. And yet it's preventable with a vaccine, and now it's also treatable. So we've come a long way from its discovery by Barry Blumberg in the 60s. He didn't know what he discovered. It was other people that showed him what it was, but it was a very important thing. It's a small double-stranded RNA virus, but it's neither double-stranded nor circular like it's said, but I'll show you that. It belongs to the family of viruses now called hepadnaviruses, hepatic DNA viruses. Its infection is restricted to humans and higher apes. This is true of most viruses. They're very species specific. High levels occur in the blood, and this is why Blumberg found it. The levels are unbelievable. There's no other virus like this in humans, where the level of virus is so high that you can detect it using what he used, which was an octralone immunodiffusion. No PCR, no radioimmunoassay, long before that. The concentrations in their blood are that of a major serum protein, like ceruloplasmin. It's spread by parenteral route, by blood transfusions. Blood is now screened for hepatitis B. It doesn't happen anymore. It's spread by sexual contact, and by maternal infant spread is a very important mode in the world. There's marked geographical variation in its incidence. It's not very common in the United States. It's very common in China. Oh boy, I'll get it right tonight. So I thought I had an electron microscope picture of hepatitis B antigen, but I couldn't find it. And nobody shows them anymore. But when you look under the electron microscope of a sample of blood with hepatitis antigen, this is what you find. You find three different types of particles. You find little spheres, lots of them, little tubular forms, and then this big thing that's called the Dane particle for Dr. Dane, who discovered it. These are incomplete surface virus particles, consist just of the envelope, the hepatitis B surface antigen. Whereas this is the intact virion that has its envelope, surface antigen, and inside of that, hepatitis B core antigen, the nucleocapsid, and inside of that, this circular small piece of DNA, and attached to that, a protein called DNA polymerase that makes DNA. And that's the particle. There's another antigen. It's funny. We talked about two antigens for many years. Then along came an antigen called EAntigen, and nobody could figure out what it is. It's a soluble antigen. It doesn't look, you can't see anything under the electron microscope. It's about 19 Daltons, and it acts very funny. It sticks to everything. Very hard to purify. What is it? We'll get to that. Boy, I keep going backwards. Here we go. So this is the genomic structure, the picture cartoon of the DNA of the hepatitis B virus. It's circular and double-stranded, but it's actually not a circle. It's two linear molecules that are held in a circle because of overlapping sequences. The one molecule is nicked, and the other molecule is incomplete. So one of the molecules of DNA is incomplete. Why is this? I don't know. It's very strange. But also, it's amazing. It's only 3,200 base pairs. That's very small. In fact, it's too small for a lot of proteins, and yet it has four large open reading frames, or four major open reading frames that produce four different proteins. The first is the blue hang here, that's the surface antigen. Then here in yellow is the core antigen, Then here in brown is as long as the polymerase. And here's an antigen called X we don't quite know the purpose of. But now the question is, how can you make two different proteins from the same DNA? How can that happen? They must be the same. No, they're different. And they're different because they're out of frame. So this virus, amazingly, has all these proteins that it makes, and they're all out of frame and they overlap. It's unbelievable, and it shows you why this virus can't mutate very much. It can a little bit, but not very much. Because when you mutate one protein, you're likely to mutate the other. Every nucleotide base is used in this virus, and usually it's used twice. So, no introns. Amazing structure. Now, it also has a lot of complexity. The surface gene, for instance, has 3 start regions. So I can start here, I can start there, I can start there. And if I start there, you get a small s. You start there, you get a medium S. You start there, you get a large S. Why does it do that? I don't know. The large S is interesting because the receptor for the hepatitis B virus is in this region, the pre-S region. It's just been discovered what the receptor is. It's a bile acid transporter, very interesting, on liver cells. But why does it have this structure? Not know. Why does it produce a lot of those small S particles that you see in serum that just are made up of the small s? Must be a reason for it to do it. Keeps doing it. So then there's the polymerase. The polymerase is multifunctional, has 2 enzyme activities. And then the core antigen also has 2 open reading frames. And that's very interesting. It has 2 start regions, one start there and one start there. If you start there, you make EAntigen. If you start at the second place, you make core antigen. How can they be different? They're the same, aren't they? And I'll show you why they're different. Then the X antigen. A lot of papers are written about the X antigen. They don't add up to very much, I'm afraid. So, circular, partially double-stranded DNA, 4 open reading frames, replicates largely in liver, probably only in the liver, and it replicates, just to make everything else complex, it replicates through an RNA intermediate and reverse transcriptase. So that's unusual, isn't it? Here's the replicative cycle. It's quite complex. The virus gets into the liver cell with this receptor. It's uncoated. The S antigen is taken off, and the core is imported into the nucleus where the DNA is released. And remember that DNA is incomplete. And what happens is it's repaired and made into a closed, covalently closed circular DNA. And that's the DNA that allows transcription of RNA. So it's repaired. And it makes RNA molecules. It makes molecules that are basically messenger RNA and make proteins, translated into proteins, core, surface, polymerase. But it also makes a large RNA that's larger than the genome. It was around, and then a little more. And this is the pregenome by which it'll replicate. This pregenome is packaged, encapsulated into the core that's been made by the other RNAs with the polymerase inside, and that polymerase starts working on the pregenome. Now remember, the pregenome is RNA, and this polymerase is reverse transcribing it into DNA. First makes the negative DNA strand, and then it digests the pregenome, and then it makes the positive strand, but it doesn't complete the positive strand. It's incomplete, and then it's exported. And there's some kind of signal once it becomes double-stranded for the surface antigen to attach to it and be exported. So the intricacies of the pathway of the hepatitis B virus in its life cycle is very, very interesting. And it gets out in the blood. A lot of it does. So I talked about EAntigen. X-ray crystallographers have recently purified enough EAntigen to do X-ray crystallography with it. And it shows you why the EAntigen and the core antigen are different. And the reason is the EAntigen's folded and the core antigen in a different way. So it has a different tertiary structure, the EAntigen. And it's the same amino acid sequence. But the different tertiary structure means that the B cell reactivity to it's going to be different because B cells respond to structure, tertiary structure. Whereas your T cells usually respond to amino acids sequence. So your T cell responses to E and to core are going to be the same. But your B cell responses are going to be different. So, okay, the other thing is the core is made up into the virus. The E antigen is pumped out into the serum. It's secreted. It's got this, it has this little tag of pre-core on it. It's called pre-core. And that has a signal sequence to be exported into the serum. It also has this, the reason it folds, it has a sulfhydryl group there that folds over on itself, whereas core doesn't have that. So why does the E antigen get out in the serum? What is it doing? Well, probably what it's doing is that it is not taken up by anti-core antibody, so it circulates freely, but it does bind to the core reactive T cells. That's kind of the current somewhat glib theory, that the E antigen is a toleragen and allows chronic infection to occur. Now, the clinical component to that is that if you are infected with blood that is E positive as a child, as a baby, almost always get chronic hepatitis. If your mother has anti-E, no E, and infects you, the child gets acute hepatitis and recovers. So it seems to be a toleragen. It also crosses the placenta, so it may tolerize the child. Very interesting. How can this virus have done this? That's what I want to know. Millions and millions of years is what it is of trying. So also the pre-core region can be mutated. This pre-core region is necessary for E antigen. So here it is, the pre-core, it has a start signal there, and there's a start signal there. If you start there, you get core antigen only. If you start there, you get E antigen, which is pre-core plus part of the core. Now, if you have a mutation right here, what is that, 1896, there frequently get a mutation there, and it causes a stop codon. So you start reading through, and all of a sudden you stop, you don't make E antigen. So you have E antigen loss, it lowers infectivity, and it seems to change clinical features a bit too. Different kind of disease, the E negative chronic hepatitis B. Mutations in the core protein probably cause changes in the virulence of the virus, we think. You can find a lot of mutations there. So enough of this. If you're infected with hepatitis B virus, what are the outcomes? This is what you see in adults infected. About a third develop acute hepatitis B. They get jaundice, they get sick, and then they recover. The majority of people have an asymptomatic infection, and they may recover on their own, or a small percentage develop chronic infection. So people who have chronic hepatitis B usually have no history of acute hepatitis B. You tell them, hey, you have hepatitis B, and they say, no, I never had that. They said, yes, you did. You just didn't know it. It was asymptomatic when it came on. That's typical of this disease, that the chronic hepatitis is silent often until you have cirrhosis or something worse. A small percentage of people develop fulminant hepatitis, maybe less, well, it's less than 1%. But to those that happens to, it's very important. With chronic hepatitis B, what happens? Not everybody gets cirrhosis. Kind of the best estimates are about 30% of people who have chronic hepatitis B will end up with cirrhosis. And this is the group that is very susceptible to ending up with liver cancer. The liver cancer usually occurs in a person who's had cirrhosis for many, many years. That's the setup. A lot of people with chronic hepatitis B actually burn out and they develop an inactive carrier state where they have hepatitis B in their blood, but they have no liver disease or minimal liver disease. The biopsies are close to normal. Their enzymes are normal. The levels of virus are very low. This is the inactive carrier state. It's also kind of a mystery what that is. Some of these people may develop cancer, but it's actually not very common if they're truly inactive. So that's adults. In children, this is different. In children, in newborns, almost all of them develop chronic infection. And that's what accounts for the high rates of hepatitis B in many areas of the world. It's maternal infant transmission, newborns with it, and they develop chronic hepatitis. And then they grow up and they pass it on to their children. Sins of the mother passed on to the child. And so it's breaking that pattern that's important, and that's the hepatitis B vaccine, of course. So here's the serology of acute hepatitis B, and it's very confusing because we have all these antigens and antibodies. But basically, during the incubation period, you first get DNA in the blood, and then you get the EAntigen and surface antigen detectable, and they rise in titer, and finally you have the hepatitis with your ALT, your liver enzymes going up. Then you recover, and as you recover, the virus goes away, and you make antibodies, you make anti-core. That comes up right away. And anti-HBs you make after you recover, after surface antigen goes away, then you make anti-HBs. Okay, that's the serology. So if you go out here and you test someone, they have anti-core and anti-HBs, you can tell them you've been infected with hepatitis B virus in the past and have recovered. Okay. If you have anti-HBs alone, that might be from the vaccine. You got the hepatitis B vaccine. Okay. Here's what happens if you develop chronic hepatitis. It's really the same thing, only you have persistence of E and persistence of S. That's chronic hepatitis. And you have a little hepatitis. It's usually mild. You may not feel bad. May feel a little bit bad, but not identified. May not become jaundiced. And then it kind of settles down and it bounces around. This can go on for years. And there's virus in the blood, so you're infectious to others. And some people, they kind of burn out after a time, and this is what happens. They have surface and E-antigen in the blood, and they have chronic hepatitis, and then the immune system or something kicks in, we don't know, whatever. They have a little flare of the hepatitis, and they clear the virus, they clear the DNA, they clear E-antigen, but they remain S positive. This is what we call seroconversion, loss of E-antigen, and it's usually followed by inactive carrier state, usually, not always. And the development of anti-E. You lose E, you make anti-E. Okay, so these people look pretty healthy and they have normal liver tests. And there's a lot of mutations that occur in the hepatitis B virus. These are minor compared to like hepatitis C or HIV, but they are important. You can have mutations in the surface gene that makes the vaccine less effective. You can have mutations in the core gene that affects disease severity. And the polymerase gene is important for antiviral resistance to the antiviral agents, because all of our antiviral agents really are directed against polymerase. It's the only thing to inhibit in hepatitis B virus. That's our problem. There's not many targets. There's one enzyme. That's it. Here's a cartoon of the world with kind of a rough estimates of the frequency of the carrier state. It's changing, of course, we hope, but it's uncommon in the developed world. In the United States, probably 0.5% of people are carriers, 1 in 200. Canada, Australia, and Europe. In lesser developed areas of the world, like in former Soviet Union, it's higher. And in the red areas, the endemic areas for hepatitis B in sub-Saharan Africa, and particularly in Southeast Asia and China, the rates are greater than 8%. For instance, in Taiwan, when this was first studied, they found that 20% of men above the age of 20 were carriers of hepatitis B. 20%, amazing. A big problem. And they did something about it. Of course, they were the first country in the world to universally vaccinate children at birth. Not the United States, not England, Taiwan. So there are many genotypes of hepatitis B virus. This is what I used to study in the lab. It doesn't turn out to be very interesting, but genotype A is the genotype in the United States. It's A because it was described first. Genotypes B and C are in Asian genotypes. Genotypes D is Mediterranean and Middle East. E is common in Africa. F is found in Polynesia and Central and South America. You could map kind of migrations of humans into the Americas with genotyping. And genotype G I'm not going to talk about. That's too confusing to explain. So here's a study that was done in the United States to find out what is the cause of chronic liver disease. What's the frequency of this versus that? It was done in the year 2000. It was funded by us and done by the CDC. And the majority of newly diagnosed chronic liver disease is hepatitis C. That's the big kahuna. Alcohol, of course, 30 or 40 years ago when we were in medical school, you assumed if you had cirrhosis, you were alcoholic. It's not true at all. There are other causes. This is non-alcoholic steatohepatitis. It looks like alcoholic liver disease, but the people don't drink. It's metabolic. And here's hepatitis B. It represents in the United States about 5% of chronic hepatitis. Long-term complications of chronic hepatitis B can be dire, cirrhosis, and hepatocellular carcinoma. Now, these are rare extrahepatic manifestations. So, this slide is getting old, but 10 years ago, hepatitis B accounted for about a third of viral hepatitis. It still is about that. The thing is that all forms of viral hepatitis have gone down in the United States, amazingly. There's less than 2,000 cases of hepatitis B reported in the United States last year. Lowest rates ever reported. There's been a decline in the incidence of over 80%, and now it's over 90%. Hepatitis B is more common in men than women. Women tend to recover from this disease. More common in African Americans. The highest rates of chronic hepatitis B are seen in Asians. And with our immigrant population, our clinic now is over 50% of our patients are Asians. And they acquired it usually in their country of origin. So when you talk about chronic hepatitis B and carriers, it comes in three basic forms. This is a little bit of a simplification, but there's EAntigen, chronic hepatitis B, E positive. They have high levels of virus. There's the EAntigen negative variety that have that mutation. They can have bad disease, but they have lower levels of virus. They're easier to treat. Then there's the inactive carrier state, low levels of virus, and usually normal liver enzymes. We usually don't treat those people. How is it spread? These are the modes. The major mode today in America is injection drug use. Vaccine. This was developed in the 1970s, and the first vaccine was approved in 1981. It's a purified, non-infectious surface antigen you're giving people, and they make antibody to it. The initial vaccine was made from plasma, made everybody very nervous because if you know what that year was, that was the year that AIDS struck, or became evident in America. So there was a lot of worry about this vaccine that was made from plasma, and along came the recombinant vaccines a few years later. I believe hepatitis B was the first human recombinant vaccine. Highly effective, very safe. You give an initial dose and then a booster at 1 in 6, so 3 doses. It's recommended for all newborns, usually the first dose before they leave the hospital, a month later and 6 months. It's recommended that school children have documentation that they received it, and there's catch-up in adolescents. And it's only recommended in adults in high-risk groups. This is problematic because it's very hard to to get to those high-risk groups, and many people don't like to admit their high-risk behaviors to doctors. The vaccine should just be used more often. Here's the epidemiology, the frequency of hepatitis B in the United States, and I'm afraid I couldn't find an update of the slide today, but the rates were the highest in the 1980s and subsequently came down, and now they're about here. And these are steps that were taken by this country, public health, to do something about it. This was very important. Surface antigen screening of all pregnant women, and then routine infant immunization. OSHA rule has to do with screening people who work with blood and blood products, routine vaccination, adolescents. So hepatitis B is a preventable disease, and the development of hepatitis B vaccine was one of the great achievements of the 20th century medicine, frankly. Think about it. Cure, prevent cancer, cirrhosis. Amazing. Just takes a long time for it to get there. Well, it's already been shown to have decreased the rates of hepatocellular carcinoma in Taiwan, for instance. The question is, why do we continue to have new cases? Every case of acute hepatitis B is a failure of our medical care system. Who is that? Anybody know who that is? Sun Yatsen, the father of modern China. He's a doctor. You know that? He was in the first graduating class at the University of Hong Kong Medical School, the first medical school in the Eastern Hemisphere. Well, Sun Yat-sen was the father of modern China, and he died at 1925. And if you do your math, you'll see that he died when he was 58 years old. You know, Asians usually live longer than that. And what did he die of? He died of hepatocellular carcinoma. So if Sun Yat-sen did not have hepatitis B, I think the history of the modern world, the history of modern China, and the history of the world would be quite different. Because when he died, he had two lieutenants. One was named Chiang Kai-shek, and the other was named Mao Zedong. And he managed to keep those people together, just managed. But when he died, of course, that fell apart. So therapy of hepatitis B, vaccine is really the approach to this disease, but there are some people that already have it, and it is a treatable disease. Now, who should be treated? Well, we base the treatment on both the presence of the virus, at least this amount, and the presence of disease. So the liver enzymes elevated. And those are the three forms. So here's the types of treatments we have now. The old treatment, the initial treatment that we used to use was interferon. And it was pretty good when it worked, but it didn't work but about 30% of the time. These drugs have come along. These are oral antiviral agents. They really grew out of the AIDS research, the epidemic of AIDS research. Yeah, that occurred. Really put a lot of effort into antiviral agents, and these agents, many of them are used for HIV. Lamivudine, tenofovir is the #1 agent used in HIV today, and it is the best drug for hepatitis B as well. So these agents are now licensed for hepatitis B. They're given once a day. They have minimal side effects, and they're fairly potent. They belong to 3 classes of nucleoside analogues. The most potent and the ones with the highest barrier to resistance, which is the problem, are tenofovir and entecavir. These other agents, I'm afraid, are going to be history, I suspect, because of the problems of resistance that you get with them. Not in everybody, but in a high percentage. So here's an example. This is interferon. This is what interferon does when it works well. Course of interferon. In the middle of the course, you have a flare of hepatitis, and you lose EAntigen, make anti-E. And this person, 2 years later, they became surface antigen negative. Well, this is a very nice response, but it doesn't happen often enough. And interferon is very difficult to tolerate. Lamivudine was the first nucleoside analog to come along. It's an oral drug once a day, very potent. Here's an example. This man was a surgeon, actually. Had rip-roaring hepatitis B with very high enzymes, high levels of virus. Put him on lamivudine, and within 2 months, the virus is undetectable. His enzymes are normal, and they stayed that way. After a year and a half, he loses E. His liver biopsies improve remarkably at 4 years, and it looks great. All right? This is isn't the end of the story, unfortunately. Here's a continuation of his slide. After his liver biopsy at 4 years, a year later, he had a breakthrough. His enzymes go way up. His virus goes right back. And he's still on therapy. He's developed antiviral resistance and breakthrough. And this can be the disease can be very nasty. So what do you do there? The explanation is a mutation in the polymerase gene that converts this catalytic site where you take up the nucleoside to one that doesn't take up lamivudine. You switch him to tenofovir, a more potent agent with less problem with resistance, and again he responds the same way with loss of DNA and normal ALT. He's been on therapy 10 years. 10 years. Still surface antigen positive. He lost EAntigen. Now, you ask, can I stop therapy now? Is it time to stop? I don't know. Some people actually on these drugs lose S antigen, like this man who lost S antigen and didn't have antibody. We gave him a vaccine. He made anti-H. We were able to stop therapy, and he hasn't relapsed. That happens. Another example of that. Of people with long-term therapy, 10 years of therapy, he finally clears surface antigen. But what we did recently was we decided that these people have been on therapy for 8 and 10 years. Do they still need it? Let's take them off and see what happens. And here's what happened. This man was on a combination of lamivudine and Decipher, had a wonderful response, lost E, and then for 4 or 5 years had no detectable virus, normal enzymes, no E, but still had surface antigen. It was withdrawn, and then, this is months. 1 month later, he has virus in the blood. 2 months later, his enzymes are up. 3 months later, he's got a severe flare of hepatitis. And he's EAntigen positive again. Yikes. So, looks like you have to keep people on therapy a long time. Here's data from industry about the rates of loss of S with therapy. With long-term therapy, you start going up, and it keeps going up, but not 100%. So the disease is not curable. It's suppressible. And long-term therapy is what we now use, but the costs are considerable. Safety of long-term treatment remains an issue. It doesn't seem to be a big issue, but it's a concern. And the efficacy of this approach is yet to be shown, although it's been shown fairly well. Here are the costs of a year of these drugs. The more potent ones cost $5,000 to $8,000 a year. So what we need are a better understanding of the pathogenesis of the disease. We need to show the safety and efficacy of long-term therapy. Interferon still hasn't been thrown out. We need newer agents with different sites of action where we can clear a surface antigen more frequently. So here's a group that I work with, the liver diseases branch, and we take in visitors, quite a few, like this guy here comes and visits and it's this group that makes all this work worthwhile. Thanks.
Okay, thank you, Jay. That was wonderful. We have time for some questions, please speak up. You mind?
Oh, I'm sorry.
Hi, my name is Maddie. Thank you for the talk about that fascinating little virus. I had a question about your slide where you showed the prevalences across the world, and I was surprised to see that Hawaii also has a very low prevalence of HBV, even though they have a lot of immigration and travel between some of the endemic regions.
Which country?
Hawaii,
Hawaii?
the state of Hawaii.
The little-
It's green on your map. I thought I would expect it to be higher because of the...
It is probably higher in Hawaii, yes. Hasn't been studied very much, but one of our groups with the Hepatitis B network is in Hawaii and enroll a lot of patients.
Thank you.
Why is it so high in the Arctic area, Alaska, Canada, Greenland?
That's the Inuit population, have a high rate of hepatitis B. And they also are routinely vaccinated, and virtually new cases have disappeared from the Inuit population. Interesting, genotypes in the Inuit, it's a mixture of everything. And what it is a native hepatitis B that was there a lot, but also introduction of Western strains of virus as well.
Jay, I really enjoy your talk, and I have a question regarding the side effects of long-term treatment of only nucleoside and nucleotides analogues like tenofovir or the others, or adefovir. So is there any study that have evaluated recently the side effects of such long-term treatment? Because, there is always the question if we really need completely to drop the use of interferon, that is more curative than relying only on the suppressive effect of the nucleoside. So I wonder if you have any data, recent data on the side effects, if there are.
Yeah, well, adefovir and tenofovir can cause a renal condition with long-term therapy that we've published on, we keep getting attacked about. It causes a Fanconi-like syndrome. It's very interesting. And entecavir has not been around long enough to say, but in rats, entecavir causes cancer. So they're very high doses. They're very concerned about that and are doing what they call phase 4 surveillance for cancer in people on long term. So that's a concern with all these agents. They're nucleoside analogues, and they're messing up DNA, and you worry about that a little bit. But so far they seem to have been very safe. And the HIV experience is running out in front of us with all this, with these millions of people on these same drugs daily, and higher doses too.
Yeah, because the
So-
rate of clearance of HBs has been patients with combination therapy with the
Right. The major side effect appears to be development of antiviral resistance.
Just a quick question. So HBV is hepatotrophic. It's in the liver. I think you mentioned that there's also other areas where it causes disease. I think it was in the renal system. How does that come about? You mentioned there were extra hepatic sites where you had problems.
Well,
with
it's a little
HBV.
controversial, but it appears like stem cells, like you can find it a little bit in the spleen, a little bit in other organs. But I think it might infect them, but it's not a very efficient infection. And once you cure the liver, those are cleared. So it requires constant reinfection. That's what it looks like to me. But there are a lot of papers about finding it in the brain and everything, I don't know.
So can someone be reinfected?
Well, the truth is it's a DNA virus. And the sad story about DNA viruses are forever. And people infected with hepatitis B have the virus in their body forever. And it can reactivate, actually. So you can get someone with anti-HBs and reactivate it if you suppress their immune system. This is really shown with bone marrow transplantation. Wow, what an experiment in nature. Hepatitis B comes roaring back. And everybody thought it was from the bone marrow that the person got, but no, it's their own indigenous. Hepatitis B is reactivated because you've taken away their immune system. So reinfection, the question of whether it's reinfection or reactivation, and it appears usually to be reactivation.
Will the anti-surface antigen and antibody exist in the blood at the same time or in the same person? Or? With the antibody and antigen.
Yes, a percentage of people have hepatitis B antigen, and they have low levels of antibody too. What they have is antibodies to strains of hepatitis B that they don't have. So other genotypes is usually what that is. It's a funny phenomenon. I've never quite understood it. And it doesn't seem to have much clinical significance.
So will the hepatitis B be cured? Is a disease free or is cured?
A disease of what?
Free, and no more hepatitis B. I mean,
no, you still see it in this country, unfortunately, because adults are not vaccinated. This is a sexually transmitted disease, so if you had teenage sons, you should vaccinate, or teenage daughters. You never know what kind of exposure there will be, and it's not. We had a case in the clinical center of a young woman with lupus erythematosus who came in with acute hepatitis B. And she was from Cambodia. I said, well, this is strange. Well, they said, well, we had screened her and she was negative for hepatitis B. Well, that was the problem. She had managed to get to adulthood and was negative for hepatitis B, and she was Cambodian, and she had a Cambodian boyfriend who was EAntigen positive. So I think that more use of hepatitis B vaccine should be encouraged. Hate to see that when it occurs. And so the question is, what happened to the person with lupus? They recovered because they have a heightened immune system, and she resolved her infection on her own. We thought for sure she would develop chronic infection.
So how valuable are the animal models of hepatinoviruses, the woodchuck and ducks and squirrels?
Those are different hepatinoviruses. So they're different.
But are they valuable in terms of understanding either mechanisms of tissue damage or interaction with the immune system?
Not very much. There's a duck virus. There are avian viruses, and there are rodent viruses, I guess. Woodchucks are rodents, right? And they're a bit different from the human virus. The difficulty with studying them is you've got to set up all kinds of immune assays that detect the cytokines and all the types of things, T cells and B cells in these animals, and it becomes very expensive. The animal models, they tend not to have quite the same type of disease. The woodchuck model was very interesting because woodchucks would get a major cause of death in them, like 80% would die of liver cancer. And even the ones that recovered from acute woodchuck virus and had antibody, no virus, would develop cancer, which is kind of scary as well. But with a lot of cancer, not very much cirrhosis in the woodchuck. And these are woodchucks that you find out along the street here. The Eastern American woodchuck has this virus.
And I had a question about isolated core antibody. So I know it can mean one of four things, but particularly so the window period of false positive, but also for people who might have actually lost the Hep B surface antibody, and I want to know how that comes about. I guess it happens more in immunocompromised patients, and then people who might also have low-level viremia, but just, I guess, lost the Hep B surface antigen. How does that, with the latter two instances, how do you explain that?
Right. So yeah, so some people, after they recover from hepatitis B, don't make anti-HBs. The surface antigen is a lousy antigen. It's hard to stimulate antibody. So some people just don't make anti-HBs. Some people make it and then they lose it. And these people have anti-core, which is a very good antigen, makes good antibody without anti. And so it's a marker of infection, and actually now blood in America is all screened for anti-core and picks up people who might have low levels of virus without detectable amounts of surface antigen, that phenomenon as well. The surface antigen is a very poor immunogen. It's very hard to raise antibody. You have to repeatedly inoculate the guinea pig or the rabbit. And this is why Blumberg discovered it, okay? He discovered it not because he had an Australia aborigine. He discovered it because of the antibody was from a hemophilic who was taken care of in the clinical center of the NIH. He went around and got all the hemophilic serum because they were exposed to multiple blood transfusions, and he was looking for genetic polymorphisms. And they would make antibodies to proteins that they don't have themselves that other people have. So that was Blumberg's theory. And so he thought for years that he discovered a new polymorphism of lipoprotein or something. And those particles, he saw those particles in serum, and he thought they were lipoprotein particles.
And then the technician developed hepatitis.
Then his
if I
technician
recall.
developed hepatitis. She always used her own blood as a control. Okay? She came in one day and said, Dr. Blumberg, I've done everything, but my blood is now positive for Australia-Antigen. And she was sick with hepatitis. So she actually was the one that figured it out, that it had something to do with hepatitis B. And it's very, hepatitis B is very common in Australia Aborigines. And frankly, he didn't discover it first in Australia-Antigen. You want me to tell the story? He didn't discover it first in Australian Aborigines. He discovered it first in leukemic patients. But he liked Australia-Antigen because he could use the abbreviation AU. It means gold, right? Because the first protein that he discovered was AG, which was silver. So this was gold, but it caught the imagination that it was somehow related to the Australian Aborigines.
At any rate, it's a great story.
Yes.
Listen, thank you very much, and there'll be time for more questions for everybody. So, Michael, if you would proceed. So we've heard a lot about the virus and the clinical disease. So now you can see from the title we're going to hear more about the host and the virus.
Great, thank you. So I won't be talking about hepatitis B. I'll be talking about another DNA virus, Epstein-Barr virus. And I'll be focusing more on the host side, so how the host deals with virus infection and how specific arms of the immune response deal with specific viruses. And so I was trying to think of how to give this talk, because this is quite a different format than most of the research seminars that we give at NIH. And I was intrigued by this cartoon where you have this apparent scientist with his microscope flying in the air, and then a clinician with a syringe. It seems very dangerous to have a syringe flying out like that. And they're running towards each other like the end of most, rom-coms, where they're going to, embrace each other and have a great life together. But and I do hope that's true, and I think that's in essence what this building is in many respects all about. But I think that there's also another possibility, and that is that students who are at NIH or postdocs who are learning about disease in an intimate way, but interested in the fundamental, demystifying the fundamental mechanisms, molecular, biological, cellular, they end up standing on the center of the bridge with a foot sort of on both sides. And I was delighted, and so we created 7 years ago the first MD PhD program in the intramural program. We've had a large number of graduate programs going back to when Dr. Varmus first opened up the NIH intramural labs to graduate students. I was delighted to see on your slide the structure of hepatitis B EAntigen. The first author, Michael DiMattia, is an NIH Oxford scholar who is one of these people trying to kind of bridge the gap in themselves and stand on the bridge. So I'll start with a little bit of biography because I think it'll set the stage for what I want to tell you. And that is, when I started college a long time ago at Johns Hopkins, I actually had the good fortune to work in the laboratories under this gentleman. His name is Victor McCusick, and he's said to be the father of human genetics, or medical genetics. And the reason why is that he began to categorize and define Mendelian disorders in humans in a very systematic way. And in fact, his categorization and his collection has continued to be expanded actually now online. And whenever you go onto the NCBI website and go to OMIM, that was created by Victor McCusick originally in book form back in the 1970s. And at Hopkins Hospital, he set up the first division of medical genetics within the Department of Medicine. And so as a young student, I worked in the laboratory, and the experiment that we were working on with collaborators at Yale were to try and map the first gene to a chromosome. And if you worked in his group, every Thursday you had to go to the clinic and listen to him and the other clinicians categorize different Mendelian disorders, and they would have Amish families come with different types of medical disorders that were clearly running in families, and they would define the mode of inheritance and categorize them as Mendelian disease. But what is absolutely astounding, at the time, everyone thought this was just state-of-the-art, incredibly exciting, really, really amazing. But we actually had no clue what the genes were that we were talking about, much less the actual pathophysiological mechanisms or molecular mechanisms. So it was really just collecting stamps in a way, in a very sophisticated way. But it was, I guess, the first baby steps towards the genomics revolution that we're now experiencing. And so I want to talk about that, and my talk will be a lot about that. But I want to point out that 40 years later, that a number of advances permit rigorous molecular analysis of human genetic disorders with a precision that matches the famous model organisms that we've become so enamored of, the worms, flies, mice, and so forth. And I just list 4 of them here. I'm sure there's some others, but these are the ones that strike me as particularly important. One is high-efficiency transfection methods. The ability to put DNA into human cells with incredible efficiency allows you to put genes in and see what happens. Conversely, RNAi, or interfering RNAs, allow you to knock down genes or reduce their expression and see what happens, at least at the cellular level. So the bottom line is, if you have a cellular phenotype in a patient, you can tell whether or not a gene is contributing to that. It's very, very straightforward with current technology. Another advance, and I'll have a lot to say about this, is so-called next-gen or next-generation sequencing with exon capture. And I'm sure all of you have heard about sequencing the genome and how cheap it is, and I'll talk a little bit about that. But that's clearly something that's made it really much more efficient to identify genes involved in Mendelian disorders. And finally, and this is often overlooked because we're so always thinking about DNA and laboratories and so forth, and that is powerful desktop computing, computer capabilities that would be unheard of even in mainframe computers back in the day when McCusick was first putting his book together by hand, actually. So I want to tell a story about these 2 little boys that came to the clinical center with a bad virus problem. And the reason I show their picture is that, well, they're very cute, but also they're pretty normal overall. They run around like little boys do, they color, they have normal mentation. So a lot of things about them are just fine. But they have one problem, one major problem, and that is they have minor infections, and these are rather uncharacterized, so I won't really go into those too much, but one big virus problem, and that is they have sky-high levels of the Epstein-Barr virus in their blood. That's true of both brothers. And that puts them into a category of patient called chronic active EBV or chronic EBV, and it's very, very dangerous. These 2 boys are walking time bombs because there's a very high propensity to developing lymphoma if you have these levels of Epstein-Barr. So of course, the parents and the physicians were very anxious to get to the bottom of this, and because there were 2 boys in the same family, and it was present very early, it was thought that it could be a genetic problem. Now, when we worked them up from an immunological perspective, we wanted to figure out whether there was something going on with their antiviral immunity. And in general, if you just want to very simplistically break down the entire immune response, there's sort of 2 ways that the body deals with viruses. And you've heard a little bit about at least one of them in the last talk. And that is, you can make antibodies against it. And this shows a little virus particle. This actually happens to be of a icosahedral cold virus. But the antibodies are these little gray things, and they can bind to it, and they can mess up its ability to interact with its receptor and basically prevent it from mounting an efficient infection. And then but if cells do get infected, then you've got another arm of the immune system in which the virally infected cells can actually be killed. And there's 2 classes of cells that can do this. One, which I'll have something to say about later, natural killer cells that react really quickly and efficiently, but they're rather nonspecific. They're the so-called innate immune cells. And then there's other ones that are much more specific for the given virus and elicited by immunization, and these are the cytotoxic T cells, so-called CD8 cells. And these will recognize either specific receptors on the surface of the cell that are turned up when the cell gets virus infected that NK cells can react against, and I'll talk a little bit about that, or antigens from the virus itself, which are presented on MHC molecules and then are attacked by the cytolytic T cells. And so these would be the kinds of things that could go wrong to predispose these boys to have this. And so we studied these mechanisms, and I'm just going to throw out some data that kind of boil the problem down to one issue. And that is when you looked at one particular receptor on natural killer cells called NKG2D, which is known to be involved in antiviral responses, and also it's thought to have some role in anti-tumor responses, compared to all the other receptors in which the blue, which is the patients, and the red, which is normals, which look pretty comparable, there was clearly a low level of NKG2D in these boys. There's no doubt about it. When you looked at the ability to kill through the NKG2D receptor, you could see it was really low compared to normal control. So of all the things that could have gone wrong, this is the thing that did go wrong. But we didn't know why it went wrong, and we wanted to get to the bottom of that. And remember, this is all in the backdrop that this is some sort of a genetic problem in this family. So now I'm going to forget about those boys for about 10 slides and just tell you about genomics, because I think this is something that is useful to know and to think about. And so this is something I think we've all been barraged with in the lay press, that in 2003 the human genome sequence was announced. Actually, it's a draft sequence. It's still actually not completely complete. But it was enough to feel like we actually had kind of gotten there. And you can see a lot of zeros after this. It cost a lot of money to do that. There were centers all over the world to assemble this sequence. And in a way, you could look back and say, gosh, that's kind of crazy. Why did we spend so much money to get one genome when now we're cranking them out by the thousands? And yet it was that event, getting the first one, getting there, that really catalyzed the fact that people said, wow, now that we can do that, let's try and do it faster, simpler, cheaper. And so now we're to the point where almost, I would say, we are to the point where we can do it for $1,000 in 2013, 10 years later. And you can see this is a curve of the cost of a sequencing per genome here. And this is the line here. You can't read it because it's too small. I apologize for that. But this is Moore's law, the reduction in size for a number of components that was the famous algorithm that is true of computer systems. And you can see, wow, we're way below that line with human genome sequencing technology. We can do it really, really cheaply compared to what it used to cost. But the other part about it is that the efficiency of doing it is much, much greater, so that you can do it essentially on the benchtop now. And so this is, again, a little bit of a personal biographical vignette. This, although you can't read it, it's kind of blurry, was a paper that I was first author on in 1984, in which I wanted to identify the nucleotide sequence of a surface antigen of trypanosomes. Trypanosomes are the parasites that cause sleeping sickness. And we were thinking about trying to develop a vaccine. And the cDNA encoding this surface code protein was 1,600 base pairs, 1,600 nucleotides long. And by sequencing it by hand took me 9 months in the laboratory of someone who knew how to do this extremely well, using 80-centimeter gels and reactions, individual reactions done by hand. And you can see the experiment I'm going to talk to you about later on these two boys that I've introduced to you. In 2010, with a 1 to 2 week prep time, 48-hour run, we generated 4 billion base pairs of quality controlled sequence. There's actually more base pairs than that generated, but these are the ones that met the quality controls. So if you do the math, the time factor reduction and the yield factor increase, you can see the improvement in time per base pair is roughly 50 million times that we've achieved now in DNA sequencing technology. So, and that's in the past 26 years. So I love to talk about this because I think we should all think about all the problems confronting us in our lives in the country in terms of energy utilization, in terms of information technology, and any technological problem you could think of, transportation, and think that at least in one venue, science has been able to deliver a 50 million times improvement in 26 years. So if 26 years from now we could have a 50 million times improvement in sustainable energy, what a different world we would all be living in. So, and here this shows science can do it. At least in this one example, they have done it. So that just gives you an idea of what's going on, why people are so excited about the genomics revolution. Now, the problem is, why are we not seeing immediate benefits? Why are we not curing every disease, solving every cancer. We've got boatloads of genomic sequence. And the problem is, in effect, the ability to generate massive amounts of data creates a problem, a bottleneck, because someone has to analyze all that data, and they have to make sense of it. We have to understand what it means. And I won't go into this issue. I could probably give a whole lecture on this. But I think the bottom line is, first of all, that in our genomes, as we'll come back to, there are a lot of changes, many, many more than classic geneticists ever could have imagined, and still have an intact human being, and we all kind of look roughly the same. But our genomes, if you were to look at them, would be vastly different going from one person to the next in this room. And I'll come back to that point. And also, how do we correlate that with health? And believe it or not, one of the biggest obstacles, in my view, to doing that has nothing to do with genetics. It has nothing to do with the NIH. It has to do with the fact that our medical records in this country are lousy. If we had all of our medical records compiled on computer systems, of course in some privacy-protected way, we could go in and do cross-correlations with every single genome we did. And the computer systems are available to do that, and they're doing that in other countries such as Finland or other Scandinavian countries where they do have good medical records, and they can do, for example, we talked about treatment with different drugs for virus infections. We have no idea when those things are approved what happens unless you do a phase 4 surveillance study. We have no idea what happens to people. We don't know if they're really working, if they're causing everyone to fall over after drinking a cup of coffee. We have really no clue unless doctors report back in a very random way to the FDA about what's going on. But it's in the medical records because bad things that happen to those people would be known. So now if we could go back and correlate that with their genomes, it would be amazing. And so this is just an article written for the New England Journal of Medicine in 2010 by Harold Varmus, our own Harold Varmus, saying 10 years on the human genome in medicine, it says physicians are still a long way off from submitting their patients' full genomes for sequencing, not because the price is high, and in fact the price isn't high anymore, but because the data are difficult to interpret. We don't really know what to do with that data yet. So what kinds of things do we do to figure out what's going on in the genome? And this just gives you a sort of a very simple summary. The kind of things that we used to do back in Victor McCusick's lab, chromosome spreads, are becoming outmoded. You can do these things all in a chip now, comparative genomic hybridization to detect changes in copy number with respect to a reference number. You get a resolution down to 1 kb. Fluorescence in situ hybridization, you can look and find parts of the genome moving around. And when you get down to nucleotide analysis, for about a decade after the human genome was sequenced, was the popularity of so-called SNP chips, where single nucleotide polymorphisms were scanned between people. I'll tell you why that's kind of gone by the boards now. But you can do either RNA sequencing of the total transcriptome. That's also supplanting microarrays, which are chips for measuring the amount of RNA, because you can just sequence all the RNAs, or DNA sequencing. And this comes in sort of two flavors, either whole or partial exome, which means just the exonic regions that encode protein, or we're moving more towards whole genome. One of the reasons why we don't do whole genome right now is that the exons are a small fraction of the total genome, and that simplifies the search for mutations that might be medically important, although ultimately we will have to do that. Acquiring data by any of these techniques is very simple. It's largely machine-driven. They're very good protocols. Any one of you in this room could reliably do any of these technologies, I'm sure. But when it comes to the data, it's very, very difficult to interpret because of the number of changes, because of the fact that all of us have many deleterious mutations in genes that are supposed to be important. And in fact, they're estimating that all of us have about 20 homozygous loss-of-function mutants in our genome, and genes that we think are there for a reason, and these are healthy people walking around. So the question is, how do you tell what's a bad mutation and what's an innocuous mutation? And that's a very difficult thing to do. Now, one of the things that has been remarkable over the past, I would say, 3 or 4 years since whole exome sequencing and whole genome sequencing has become more commonplace is the whole idea of genomic variation has radically changed since back in the early 2000s. So the original concept, which was championed by the leaders of the Genome Project, was that common diseases would have common variants. And so that if we looked at the HapMap, so-called the haplotypes of SNPs or GWAS studies, we could see that we would correlate these SNPs in studies that were widely funded by the NIH, and therefore understand why people get diabetes or why people get mental retardation. And the bottom line is, although this gave us a lot of correlations with genes, the problem is many of these SNPs were not in any way identifiably associated with a change in gene function. It was very difficult to find out what these changes in nucleotides were actually doing. The effect on genes were not clear. Many were actually not in genes. They were in regions between genes or in introns, and it was very, very tough to figure out what was going on. Now, that wasn't the biggest problem because, we're scientists. We figure out hard problems. That's what we do for a living. But the problem is that when you totaled all these up in any given disease, type 1 diabetes, lupus, whatever correlation you want, the frequency of these was very, very low, and the amount that they could account for the genetic variation was exceedingly low. So the bottom line is any given SNP you looked at didn't really have much influence over whether or not someone got that disease. And on top of that, there was very few medically actionable variants. So a variant, you could point to that and say, okay, if that protein is mutated in its active site, that's going to cause a predictable effect on phenotype, and we can now try to do something about that. So the International HapMap is pretty much a bust. All these companies that are now, analyzing DNA, you can send them in your spit in a little tube and send it off to a company, and they'll tell you whether you have the Parkinson's gene or the mental retardation gene or any of these genes. It's completely bogus. These are SNPs analysis that they're doing, and the likelihood, first of all, it's correlation, so the idea that it's causal is not proven for the vast majority of these SNPs. And secondly, whether they're going to have any influence on your life or your family's life is very, very low, unless they're testing for specific mutations and specific genes that cause disease like Huntington's disease or cystic fibrosis. And those are usually done by medical professionals, not by mail-in companies like 23andMe. So this whole approach to genomics is really been not the salvation of our health that we thought it was. Why? Because of the vastness of genetic variation. This is what I'm going to talk about in the next few slides. The bottom line is classical genetic estimates that there were hundreds or thousands of unique variants per individual. So they would fall in important places and they would have important effects. The bottom line is that whole genome sequencing or even whole exome sequencing shows that we have millions of single nucleotide variants, insertion deletions, and copy number variations compared with any given reference sequence. So what's going on inside your genomes is a whole cornucopia of molecular change. And now, not only the fact that we don't have good medical records, but the point is in any given individual, we really don't know what most of those are doing to your ultimate phenotype. I'm certain, as I'm standing here, they're going to have some effect on your phenotype. And as we go through our lives, some of us are going to suffer more from certain illnesses than others. We're going to die from different illnesses than others. And those will have some impact on that. But how they do that is a very complicated question. And in fact, we don't even really talk about SNPs because there's so many of these variants. We call them SNVs, single nucleotide variants, because a polymorphism is defined as in 5% of the population. Most of the SNVs in all of you are very rare. They're not going to be present in 5% of the population. So now we have to deal with this genetic complexity because we could never observe it before, but now we can, and we have to understand it. So how much variation is there? I alluded to this a little bit, but let me just review this. Whole genomes show much more rare and private variation than expected. SNVs, about 3.5 million per genome, 1 per kilobase. Copy number variations, at least 1,000 per genome. Most of these are not in the population from which the individual comes from. So the idea that all Ashkenazi Jews are going to have the same variations is not true. They're going to have some shared variations, but they're going to have a lot that have never been seen before. In fact, we're still so early in the game that each whole genome sequence is still yielding hundreds of thousands of single nucleotide variants that have never been seen before. So now, why do I have a picture of Stone Age people here? Why do I do it? Because I want to introduce the idea of clan genomics. Your own clan are the people that are going to have nucleotide variations that are similar to you. And when you get beyond your clan, they're not going to be the same. And so So with the human population explosion, older fathers, relaxed selection, and even medical intervention, we're keeping a lot of genetic variation in our species that is now going to be something we have to deal with as medical geneticists. So Lupsky wrote a very influential and very logical review in Cell in 2011. I'd encourage everyone to read it, where he talks about this concept of clan genomics, that all of us are going to be most like our siblings, our parents, and our children. And when you get beyond that little group, you don't. And so if you look at this clan, you can probably see somebody here that looks kind of familiar. This is way back when I was in Victor McCusick's lab. Why is that? Because through selection, if the variants have a significant enough clinical impact, as they go through, spread throughout the population, they'll be eliminated because of the effects that they have. And so that those variations, when they first occur, in genomes that are shared, largely shared between the members of your clan, are going to be present. And that the only ones that get left when you get out in the whole population after many generations are the ones that have very small effects. So the ancient and common variants are the ones that really are not going to have a big impact, and that's exactly what GWAS studies and studies of SNPs prove to us, that you get variants that have very little impact on disease. We did the experiment, and that's what it showed. So let's come back to our boys. So needless to say, based on everything I've told you, we were anxious to do whole exome sequencing. We actually had a molecular effect in their cells that we could trace to see whether the gene was in fact a true gene. We found that in fact both boys shared an X-linked gene mutation, a 10 base pair deletion, in a gene called MAGT1. And it was a deleterious mutation. Remember, boys only have one X chromosome, so they only had one allele, and that was mutant, and it eliminates this protein in their cells. So that's very interesting, but what the heck is MAGT1? And to our astonishment, MAGT1 was a magnesium transporter. Had never been implicated in any way, shape, or form in immunological phenomena. In fact, there were only 2 papers in the literature. One was a paper in zebrafish where it had been knocked out, was embryonic lethal, and the other is a study in which they studied it biophysically and could show that it could conduct, highly specifically, a magnesium flux across the cell surface membrane. So the question is, now, how does a magnesium transporter affect cytotoxic T and K function, and by extension, the susceptibility to Epstein-Barr virus? And I don't have time to really get into the nuts and bolts of the research story, but I'll just tell you that your cells, unbeknownst to us and a lot of other people, have sort of 2 kinds of pools of magnesium that are different than the pool of magnesium that we all learned about in biochemistry. We all learned magnesium is critical in our cells because it binds to ATP. It's required for a whole lot of enzymes that it binds to very tightly. It binds to membranes. It binds to chlorophyll. And it acts as a cofactor, allows them to do their job. But in addition to that, there's also a pool of free magnesium in the cell, about 1 millimolar that we found. And this is true in most cells in the body. It's certainly true. This is an example of lymphocytes here. And for T cells, although I won't have time to really get into this, there's also a TCR-gated magnesium flux, very similar to the calcium flux. In fact, it precedes the calcium flux, and the calcium flux depends on having this magnesium flux across the membrane. And you can see both of these pools in the patient, one of the boys here, are lacking. This one's reduced significantly, and this flux is completely abrogated. And that has effects on the boy's cells. Now, we went through and, with our colleagues at NIH, once we had identified this mutation, and were able to find other examples of individuals that had a similar kind of a phenotype, meaning pretty mild, normal-looking people that had sky-high levels of EBV, and we thought they would have a susceptibility of lymphoma. And you can see, in fact, similar to the problem with hepatitis B, with EBV, you get lymphoid malignancies, which is the target cell that it infects. And you can see all the older patients that we found that had mutations in this gene had high levels of EBV, but had developed lymphoma. In fact, 2 of them had developed 2 different independent primary lymphomas after one had been treated. So the question is now, how do we relate the high EBV to magnesium? And this really tickled the little boys that had this disease. We called it X-linked magnesium deficiency with chronic EBV infection, neoplasia, or X-men disease. So these are the first X-men here in this group. So the way that we came about this was sort of in a roundabout way. We were confused about how you would explain a constitutive lack of a receptor on these immune cells. It certainly didn't seem to be related to the flux, because if you triggered this NKG2D receptor that I told you was messed up, it didn't cause a flux of magnesium. So we thought maybe it's the basal-free magnesium. And when we went back to the literature, we found out that there are actually 22 different magnesium transporters in eukaryotic cells. The distribution of those and the function of almost all of those is completely unknown. There's only one, TRPM6, which is the reclamation receptor for magnesium in the gut and in the distal renal tubule, which has a significant literature on it. So we thought maybe one of these other transporters could substitute for this important function of keeping the basal level of magnesium high. And so the experiment that we did was simply to raise the amount of magnesium in the extracellular fluid above the normal level, which is about 1 millimolar in tissue culture media, to see what would happen to the patient's cells, and then also at the same time measure NKG2D on the appropriate cell types, which were, if you remember, NK cells and CD8 T cells. And so what you can see here is that, in fact, when you do that, as you raise the amount of magnesium in the extracellular fluid, you actually can increase the level of intracellular free magnesium, and you can also actually increase the level of NKG2D. So this was really an astonishing result, because it told us that the lack of that surface receptor was due to the low level, or the diminished, artificially diminished level of magnesium inside those cells. So it depended on that. And I won't get into the nitty-gritty of why that's true. It has something to do with the glycosylation. It's a heavily glycosylated surface protein, and that doesn't work properly when the cells don't have enough free magnesium in the cell. But what was gratifying is that when you restore magnesium on these cells in the culture dish, you also find that the killing function of these cells against EBV-infected cells goes up. So that gives a very nice model to think about and understand how this was actually working in these patients from the human genetic analysis of these hosts for the virus. And one of the amazing things is that being at the NIH, you can actually do something about that. And we did, and that is we brought the boys into the clinical center and we started giving them more magnesium. Either we did different ways to do it, by IV or by oral supplementation. In fact, they're on oral supplementation right now. And again, I won't have time to go into all the data, but it was incredibly exciting that within a few days of starting oral supplementation, you can actually see the level of NKG2D going up on CD8 T cells and on NK cells. In the patient, it doesn't reach the normal level, but it goes up substantially. And you can see by using a fluorescence in situ hybridization assay done in Jeff Cohen's lab, the number or the fraction of EBV positive cells going down. So actually, in a human being, we can show that there's a correlation between this basal magnesium, between the expression of just one, and there may be others, but at least one that we can identify, key cytotoxic cell activating receptor, and the control of this virus. Now, is this the only thing that helps control EBV in our body? I doubt it. I'm sure there are other things, but the fact that one seems to be pretty darn important, I think, gives us an insight into why our immune systems are so complicated, because we deal with a lot of different kinds of viruses, a lot of different kinds of pathogens. And so to have a lot of different tools in the shed that you can use that might be particularly good for different viruses seems to be a very good evolutionary development for humans. So I think the idea of studying Mendelian disorders to understand host virus interactions and understand immunodeficiency is a very powerful approach. Obviously, it would have been much tougher to do this back in McCusick's era, but I guess he sort of started us down the road to getting to where we are now. And with the technological developments that we now have in hand, we can do this successfully and reproducibly in different types of disorders that we're confronted with when patients come to the clinical center for analysis. And so what did high-powered DNA sequencing combined with medical genetics teach us? Well, it taught us the pathogenesis of X-Men disease. It gave us a new fundamental understanding of magnesium that we didn't have before. It gave us ideas on how to treat the disease. And I want to introduce a note of caution here. I don't want to suggest that these early results that we have with these patients mean that we will be able to treat them effectively and rid them of EBV or even significantly lower EBV. We've seen bumps, but whether this is going to be the ultimate treatment, I would hesitate before making that claim. And then finally, it gives us insight into the physiological and immunological role of a gene that was hardly studied previously, at least not in humans. So with that, I'll just thank the real X-Men of the story, Feng-Yen Li and Ben Chen de Lalonde, who did a lot of the work from the first gene identification all the way through to analysis of the patients. They were helped by a large cast of really talented people in the lab doing all sorts of different things. We also collaborate with Helen Su's lab in the clinical center, who's been involved in our genetics program. And of course, the NK experts in NIAID, Eric Wong, Sumi Rajagopalan, and others around campus, Danny Dewick, Gulbu Uzel, who actually is the physician for these patients and has been great to work with on the clinical side. Jeff Cohen, who of course is our EBV specialist, and a number of people at other centers. So I'll stop there and take any questions.
Thank you. Thank you, Michael. That was very exciting. Do we have some questions or comments while we're getting
Yes.
a question? Here you are.
Thank you. I have two questions. The first question is about your NK cells, except the NKG2D. Did you measure some inhibitory, receptors? For example, NKG2A or
In
whatever?
terms of surface expression, every other thing we measured was unchanged in the boys.
Okay. So, and after, feeding the boys with the magnesium, right? The NK cell number increased. Have you measured the functional of the NK cells?
Yes.
So they increased.
The function is increased. I didn't show all the data just for
Okay.
lack of time.
So the second question is about the B cells. So you show the low intensity of virus in the B cells, right?
Yes.
Is the infected B cell number reduced, or they are just have a lower load of virus particles within the cells?
The data, the FISH is kind of a nice technology because it allows you to evaluate both of those things. And what Jeff and his colleagues saw was a reduction in the fraction of EBV infected cells and a reduction in the intensity of fluorescence in the nucleus of the ones that were left over. So the ones that had lower levels of virus seemed to be the ones left behind, at least at that short time course. We now have a new flow cytometric assay that's just been developed in the lab by Ben and Ping Zhang, and they're actually going to now look very carefully, and they can run thousands of cells, on the flow cytometer and get an idea of the fluorescence.
I'm wondering whether it's interesting to measure the absolute B cell number in the blood.
Oh, the absolute B cell number in these boys is slightly higher than normal. They're not low in B cells. They're a little bit higher than normal.
So Michael, which came first, the genomic sequencing or the measurement of magnesium in the blood?
Oh, the genomic sequence. We would never have thought about looking at magnesium in a million years without, the identification of a magnesium channel. Yeah.
Very interesting study. I wonder just if you had also the possibility to investigate the humoral, part in these two kids, all the antibodies, as you showed at the beginning. Or
Yeah, the
you just
vaccination
focus-
responses were pretty good.
Were pretty
I think
good.
they might have been slightly hypogam on a couple of determinations, but generally they're pretty much in the normal range.
So
So
they
it
were
doesn't
up-
look like a big deficit of the humoral
Okay.
immune system. Now, we haven't gone back and looked at EBV-specific
Exactly. That's
antibody
what I was
stuff.
thinking.
I
yeah.
suppose we could do that. But
Okay, I was just thinking about this. Thanks.
By the way, I should mention, for those of you who don't know, all of the PowerPoints, references, some CVs, and a few notes and whatnot are on our website, and you can download them, look at them, study them, and so forth. Are there other questions? So if one were to go off the deep end, considering that we're started out with hepatitis B, What might be your thoughts about how one might confront some of the issues that Jay brought up, different clinical phenotypes, and is that all due to the virus, or is it worth exploring for less than $1,000?
I think, you know, you don't want to go out and just sequence genomes randomly of virus-infected people. I mean, a lot depends on the virus biology. Viruses evolve much faster than people, so they're going to be susceptible to genetic change more rapidly. But I think that if you see virus susceptibility issues in a family, so if Jay were to say, hey, Mike, I've got, you know, this, you know, huge extended multiplex family where it seems that some people can get rid of the virus and some people can't, then that's the sort of thing where looking at host factors might be very revealing. And I think, you know, the idea that I want to leave everyone with is that if you look at mental retardation, which is kind of a common thing in the population, that rather than think of a common variant that's going to account for all those cases, in each of those individuals with mental retardation, there may be a different, very severely affecting a mutation or variant that's causing it in that individual, and that's going to be different than the next case. And it's only when we discover all those and put them together with systems biology and bioinformatics will we begin to see the pathways that all have to be intact for normal mental processes.
What about the differences in, which Jay mentioned, in different racial groups, different manifestation, you know, I guess it's not just frequency of the disease, it's severity and phenotype, which varies.
I would think that would be more to their food choices, sanitation, things that are not related to their genetics. Because again, remember, when you get beyond the clan, no matter what racial or ethnic group you are, those differences are going to be able to become more and more minor as you go broader out through the population. So there may be a few scattered susceptibility genes for hepatitis in Asian people, but I think probably it relates more to how people live. And if you're living in a crowded city in India where there's a bad sanitation, you might be much more likely to pick up a virus like this. Or if you're an IV drug user, that's not your genotype that's going to.
Well, there's certainly apparently racial differences in hepatitis B and frequency and so forth. But part of the complexity is like genotype could account for a lot of it, the Asian genotypes versus the African. But the epidemiology of hepatitis B is very different in Asians than in Africa. And in Africa, the rates of cancer strikes, the average age is like 40, young men, you know, minors. It was a major cause of death in the mines in South Africa. Whereas in China, it's a little bit more like the United States. The cancer is occurring in 55, 65-year-olds. So those types of things are clues to follow up on, but I agree it becomes very complex.
Yeah. I mean, I'm sure there's some genetic component, but it wouldn't be the first thing I'd think of.
Okay, well, listen, thank you both.
You're welcome.
Very much.
My pleasure.
Very exciting. So next week is sexually transmitted diseases.